Thank you very much. I'd like to thank both Turkson and the organizers of this meeting for inviting me to do the presentation. I will focus on Africa generally rather than South Africa. And to focus on personalized medicine or precision medicine, we must realize that we are all unique. Our health is determined by our inherited genetic differences, but combined with lifestyle and environmental factors. So, and personalized medicine is a move away from the one-size-fits-all approach, and the treatment of patients is better, have managed better with targeted therapies to achieve the best outcome and manage the disease, knowing the predisposition of the disease. So the one-size-fits-all model assumes that all patients are the same, although they're genetically different, they're exposed to different environmental factors, but they're all given the same treatment. And so with precision medicine, obviously, as we all know, genetic analysis, environmental analysis highlights the differences so that treatment is tailored to match the individual's makeup and response so that you have more effective treatment. So defining the landscape for personalized medicine, it's not only about the genes and genomics. There is the epigenome, the modifications of DNA, modifications of proteins that bind the DNA. There is the transcriptome, the quantities of messenger RNA present in the cell. There is the proteome, the amount of protein, the modifications of proteins, the activity of proteins that are important. And very important is the microbiome, the bacteria living inside us, the bacteria living in the gut that are able to metabolize drugs, that are able to generate metabolic products that can affect disease. So the landscape for personalized medicine is fairly complex, but what we often take into account is mostly just the genome. So there's been excellent progress in many populations that have led to databases, genomic databases, that more accurately reflect the natural diversity in those populations. So these genomic discoveries have made clinical trials and medical care more successful for participants in those particular population groups where the genetic background is defined. So for example, in the Korean Genome Project, there's a population-specific variant in the gene that regulates the metabolism of some of the drugs in the body. And this is essential information for both the dosage and gauging the likelihood that the patient will respond to a particular therapy. However, shifting focus to the global south and to Africa, there are glaring deficiencies. There's an underrepresentation of certain ethnic groups in the scientific data. Human genomic databases, for example, are heavily skewed towards European populations. And this bias will continue to contribute to uneven success rates in precision medicine. So in fact, although we're going to precision medicine, we're going back to the one-size-fits-all concept by assuming that all genomes are the same. And so just to give you an idea about Africa, how big is Africa? Africa is larger than China, United States, and Europe combined. And so the genetic diversity in Africa is immense. So if you look at, based on the HLA genotyping, There are the migration out of Africa has been associated with large genetic variation. So that if you look at the blue arrow, West Africa, the Yoruban population, and you go south to Southern Africa and the Khoi and San populations, the genetic variation between those populations are different. So that um, precision medicine will not be the same for those unless you know the genetic polymorphisms and genetic makeup of those populations. So some of the considerations that we have to take into account is that currently, if one looks at the human genome databases, the African genome comprises less than 2% of the human genome database in the genetic databases. The African populations are genetically the most diverse of all populations. And the publication by Sherman et al. Showed that the Human Genome Project reference genome lacked several ancestral variants, African variants, and it's estimated that the genome would have, oh, sorry, that's a mutation there, would have 10% more, not DNA, 10% more genetic variation if the African population was included. So the H3 Africa project, H3 stands for Human Hereditary Africa, H3 Africa. In that project, they analyzed 426 genomes across 15 African countries from about 50 different ethno-linguistic groups. And they revealed 3 million unknown variants in the African population, just from that 426 genomes from 50 ethno-linguistic groups. So it is estimated that to make a meaningful contribution to capture all the genetic variation in Africa, we will have to sequence about 3 million individuals to capture all the genetic variants in Africa. So the African Academy of Sciences have funded the H3 Africa project, largely through grants by the US National Institutes of Health and the Wellcome Trust that was managed by the African Academy of Sciences. That is a 12-year project that has come to an end. Roughly 180 million US dollars was invested in that project. And that project ranged from population-based genomic studies of common disorders, cancers, and infectious diseases, etc. So it covered the whole range of genetic variation. Currently, there are also many other fellowships, the ARISE Fellowship, a grant from the European Union to fund young researchers. Under that program, 44 awards were made to young researchers in 38 African countries. And we also have the African Postdoctoral Training Initiative fellowships with funding made available by the US NIH. So there are various funding programs in place. However, a lot of these programs have their limitations. The funders are often very prescriptive in the types of studies that they will fund. So, for example, if you look at the leading causes of death in Africa, the top will be neonatal diseases, followed by respiratory infections, diarrheal diseases, and then HIV/AIDS. But most of the funders will insist on funding HIV/AIDS, malaria, and TB research, rather than the diseases at the top. So if you look at stroke, heart disease, those two combined kill more people than HIV/AIDS. But there's a low priority for funding those types of research, or research into those diseases. Um, and so going back to the genomic studies are almost exclusively based on European American genomes. So for example, the H3 Africa project data showed that a, a mutation or a variant of the cytochrome P452B6 gene called CYP2B6 star 6 affects the metabolism of ephedrins in Africans so that they have very bad side effects. With the result that people tend to skip treatment or avoid treatment because of the side effects. But that's not taken into account in the genotype. There's also some skepticism about foreign funding. For example, when the UK Wellcome Trust Sanger Institute tried to commercialize a gene chip profiling kit based on African genome, done largely through the H3 Africa project. So one has to be careful in the way one proceeds with foreign funding. Just to go to my research area, I'm focusing on cancer of the esophagus, and esophageal squamous cell carcinoma is very common in developing countries, whereas adenocarcinoma is very common in developed countries. So the adenocarcinoma is largely due to gastroesophageal reflux that occurs at the junction between the stomach and the esophagus. Squamous cell carcinoma occurs mostly in the upper esophagus. There are no early warning symptoms. So when the patient comes to the hospital, the disease has advanced and the prognosis is very poor because of the late stage of diagnosis. So if you can look at that chart, you'll see that Eastern and Southern Africa and China are the highest incidence areas for squamous cell carcinoma of the esophagus. In South Africa, if you are a black male, you have a lifetime risk of 1 in 30. So in other words, that you have 1 in 30 males have the risk of developing esophageal squamous cell carcinoma in their lifetime. It's slightly lower for females. It's about 1 in 20 for female, 1 in 40 for females, compared to 1 in 500 for white South Africans living in the same environment. So there clearly is some sort of genetic factor playing a role in susceptibility to esophageal squamous cell carcinoma. So what we did is we did whole genome sequencing on tumors, DNA isolated from tumors of squamous cell carcinoma patients. Because of budgetary constraints, we could only do it on 35 patients. We sequenced the tumor and the corresponding normal DNA, blood DNA from the same patient and compared. So you see there's some of the patients' comparison. And you can see that the red one shows you the single base substitutions. The blue shows you the double base substitutions, and the green on the end shows you insertions and deletions. And I would like you to focus on the patient second from the bottom, with a large number of single base and double base substitutions. When we did, sorry, you won't be able to read this, but it's just to get an idea. When we did mutation signatures on those patients, if you look at the second patient on the left-hand side from the top, just look at the right-hand side of the bar, you'll see a large increase. Sorry, I don't have a pointer to show you, but you'll see a large increase in the pink bars of the patients. And that patient is unique. You don't see that in any of the other patients. So the mutation signature for that patient is different. So if you look at it, there are several different types of mutation signatures, and the patient is shown on the right-hand side of this bar again, where you can see the second patient from the top, the huge pink bar, indicates what we call mutation signature A. Okay, and we don't know what that mutation signature is due to. For example, if you look at B, C, and D, you can see B is largely due to DNA mismatch repair deficiency, polymerase D1 mutations, etc. So the cause of the mutation signature is largely known, but for that patient, it's unknown. It's not been detected before in any of the other populations. So clearly there's something wrong. We need to look at a larger patient cohort to see how significant that is. But obviously we'll have to sequence many, many more hundreds of patients. So the way forward, how do we move forward? We clearly need true global collaboration. Very often collaboration these days means Africans researchers sending samples to Europe or the United States, the work is done there, and the co-author on the paper, but none of the capacity building and research is done in Africa or by African researchers. The role of African governments, as showed by Meman Chandi, that the budget, the expenditure is the highest expenditure servicing government debt, then defense, and then a whole lot of other things, and health and the research is way down at the bottom. So that needs to change. And also in terms of funding, funders need to try to coerce African governments to collaborate and contribute to funding by matching funding from governments. We have a problem with human capacity development. So most of Africa's outstanding researchers are in Europe, United States, or somewhere else. And we need to ask why. Because there's no incentive for them to come back. There's no infrastructure, there's no support when they come back. So why come back? So in order for their own progress, for their own good, it's better to stay where they are. So things need to change drastically in the African continent. The same applies to infrastructure. We need good biobanks. And more importantly, we need good governance. We need to have good oversight. There's too much corruption. And funders need to make sure that oversight mechanisms are in place and strictly enforced. So thank you very much from Cape Town. And I hope this has been useful. And thank you once again to the organizers.